Okay, so we have these plasmids, cool. How do we actually put them into a bacteria? This is just the DNA. So we have actual just plasmids isolated that we can do this to, and somehow we need to get these into a bacterial cell. So first thing I want to point out, I'm going to use this video and kind of record over it because there's really good videos, but this shows a section of frog DNA. In our lab that we would do in class, we would use jellyfish DNA, but still we have a gene of interest. In our situation, it's this GFP gene that makes the jellyfish glow. So we have this gene, but we have the whole section of DNA. So this just showed like a small portion of that section of DNA, but that's just one gene, and we have the whole DNA. So what happens is when the restriction enzyme gets mixed together with the DNA, it can actually cut it in a couple of places because you could have those same repeats, that same recognition site, several places in the DNA. And these dotted lines here actually are showing the different sites that that specific restriction enzyme can cut at. So yes, it cuts perfectly around this gene. That's what we want, great. But also it cuts here. So when I cut here and here, I'm gonna have this random piece of DNA, and it cuts here and here on this other random piece, and this piece, and this piece. So when I cut the DNA that I want, I get that one section of the gene I want. This is my gene of interest. But then I also get some random pieces of DNA. So I cut this up and I get all of those pieces. I get the gene I want and some random ones. And then I cut the plasmid. Cool. I cut the plasmid, so nice, so good. But then when I mix them together, and I actually mix all of, so the process of I cut the DNA and I cut the plasmid and I mix them all together so that they do this, because I have all of this regular random DNA and this gene, there's 3 actual plasmids that I could get. Not every single one is going to be perfectly the way that I want it like this. So some of them, yes, are going to get the gene that I want. Wonderful, wonderful. We're going to talk about what this little red thing means here in a second. But this is one of the plasmids. I mixed them all together. One of them, or a few of them, nice, actually get that gene I want. Awesome. But I'm mixing all of this together. So some of them are going to take up this random DNA. So still took up some DNA, but I don't really care about these. And then some, because they're, let me see if I can get a better visual of this. So I get some that I want, some with random DNA, and then some that just honestly close up on themselves. So this one here, I opened it up or cut it up, and then it just closed back on itself and didn't actually get any of the DNA at all. So I have these 3 potential options. A plasmid that took up the DNA I wanted, a plasmid that took up some random DNA, and a plasmid that just closed up on itself. So that's what I have here is this one. Very cool, very nice. So how do I get it into the bacteria? Well, I have a bacterial cell. Looks like this. It's unicellular and its DNA is just floating around. So it has its own plasmid. I don't actually edit the DNA of this bacteria. I just stick in another plasmid. And this will, you'll get a nice visual of this here in a second. So I have this bacteria and I heat shock it. I heat shock it, and that will cause several holes to open up within the cell membrane. And so she visualizes it here. That plasmid sneaks in via one of those holes in the cell membrane. So now the plasmid's in there. It has its own regular DNA. It can do all of its regular stuff. But then I have this new plasmid too. So its machinery, all of its ribosomes, are going to also transcribe and translate this. And this one that took up this nice cool gene that I wanted is going to glow as a result of making these proteins. So I heat up a bunch of the bacteria. They get holes in them. I mix it with the plasmids. Plasmids sneak in, and then the holes close back up because I cool it back down. So there are now 4 things that could happen. So I could get that one that was pictured here that took up my plasmid that I wanted. But remember, I have some of those random plasmids too that just have that random DNA. So some of them are going to take up the one I want. Some of them are going to take up that random one that had that random DNA. Some of them are going to take up the one that closed up on itself. And some of them aren't going to take up any at all. So some of them are going to be left like this. So now I have 4 different types of bacteria happening. So how do I figure out what I have, what I'm working with, and if it worked or not? So what I do is I take all of these and I plate them on, these are like Petri dishes. And I have one here to show you a better visual. So there's a lid, and this one is like a bowl almost, but it has a very flat bottom. This is a really old one that still has some stuff in it, but I put some agar on the bottom, and it's just basically a gel, and I mix it with nutrients, with nutrient broth, and that's just like a medium where the bacteria can grow. So I make it to where the bacteria can grow on this broth, all awesome, all good. But I also add in ampicillin. So ampicillin is an antibiotic. So break that word down, antibiotic. It's antibacteria. So antibiotics kill bacteria, and that's why you take antibiotics when you get sick with bacterial infection. But why would I do that? Why would I kill off the bacteria? Well, that brings us back to that red triangle that I said we will revisit. So all of these plasmids are going to be a little bit different, or have different options. Some of them could take up my good gene that I want. Some of them could take up random DNA. Some of them can close back up on themselves. But they all have this already. I picked a specific plasmid that has this, and it's a gene that codes for antibiotic resistance. So all of these plasmids will produce a protein that allows those bacteria to be resistant to the antibiotic. And that means that those ones that took up any of those are going to live. So anyone that transformed any one of the potential plasmids are going to live on the agar that has the antibiotic, because it has the antibiotic resistance gene, and it will live. So that's one way I can tell, like, did it get a plasmid, right? Because if that bacteria couldn't grow and it died, that means it just has its regular old DNA that's not resistant to the antibiotic. So the ones that took up nothing, these guys just die and they can't grow. All those other options can grow, and then when I look at the ones that grew, I'm going to be able to see whether or not they glow under UV light. So I have a picture here. You can see they both grew. You can't really see the colonies. The colonies are those dots. And actually, that's hundreds of bacteria just in that one little dot. It's called a colony. But you can't see it here. This one actually has some dots. But when I look at it under the UV light, this one glows and this one doesn't. So that means that this one either got the plasmid that closed up on itself because it still has the antibiotic resistant gene, so it didn't die, or the one that took up some random DNA. I don't know what that DNA does, but it doesn't cause it to glow, so it's not my gene of interest. But again, still had the antibiotic resistance, so it still lived. This one must have been the one that took up my very nice gene of interest, and now it's glowing because it's producing that protein. So cool, huh? And the ones that don't grow at all, remember again, they must have died, so they don't have that antibiotic resistance gene.